the water use efficiency and the relationship between transpiration and stomatal conductance. If you're interested in this, there is a good paper by Seep et al., 2008, called Carbon Isotopes and Water Use Efficiency in Sensitivity, that was published in Ecologia. And it talks a lot about the difference between instantaneous versus intrinsic water use efficiency. Any questions about some of the extra measurements that we can get beyond gas exchange, just pure gas exchange, stomatal conductance, and things like that? Okay, so then let us talk about what we want to do with this instrument. So hopefully now you understand how the LI 6800 hardware works, how the LI 6800 measurements work, things to keep in mind when you're taking your measurements. So let's talk about what everybody really wants to know is, and how do I take all of that stuff about the biochemistry and the physics and the electronics and everything like that and turn that into something that I can write a paper on. So let's talk about the parameters that researchers want to measure. They might want to look at net flux of CO2, which is photosynthesis or respiration, very common. They might want to look at transpiration and stomatal conductance. They may want to look at internal CO2 concentration or chloroplast CO2 concentration, electron transport rate, and velocity of carboxylation, the speed at which Rubisco is carboxylated. There are a lot of other things that researchers want to measure, but most of them are kind of in these categories in terms of looking at the net flux of CO2 or looking at the net flux of water. They might, or the CO2 internal concentrations and then chloroplast concentration. You need fluorescence to measure chloro concentration. You can do most of the others just with gas exchange, but you can also get some of them from fluorescence. So once you've decided which parameters you would like to measure, then the next thing you need to think about is what plant you want to use. And once you've decided what plant you want to use, you need to think about what you know about your plant, what you expect from your plant. If you have a C3 or a C4 plant, what is the maximum photosynthesis rate that you would expect to see? What is the CO2 compensation point? What is the optimum temperature? What is the anatomy and the carboxylating enzyme of this plant, and how does that affect what you would expect to see? This is just talking about C3, C4, and CAM. It's not going to talk about any of the intermediates or any of the plants that switch between C3 and CAM or C4 and CAM, because there are certainly a lot of those. But in general, you would probably know if you were working with a plant that was not easily fitting into the C3, C4, and CAM. Also, we're not talking about the C2 cycle, but that's fine. But think about what plant am I working with? I heard maize, and I heard wheat, and I heard some horticultural crops. So what plant are you working with? What numbers do you expect? What reality do you expect to see? And the other thing I will remind you when we're talking about knowing your plant is, I saw this on the wall of a lab that I was visiting to do a training. And it's one, I can't figure out where it's from. I have Googled it. It has not worked for me. Maybe someone just came up with it. But the principle is to never believe a measurement unless it can be substantiated with theory. So if I saw that I had a maize plant or a wheat plant, and their photosynthetic rate was wildly different from what I would expect, then I should start thinking about what could be causing this. What is the theory of this measurement, and what could be causing this number to be completely different from what I expect? So it's always a good idea to look in the literature and figure out what numbers you expect to see, whatever reason. You don't want to be in a situation where my advisor used to say, you have a nature paper or you have a problem. It's always better. It would be nice to have the nature paper, but it's usually a problem. And then, you need to start thinking about that measurement versus that theory. This is how the instrument works. This is how the plant works. Do I expect these numbers that I'm seeing? If not, why? And if once you're trying to get into the why, call us or email us, call tech support, say, this is the measurement I'm getting. This, I don't understand it. Maybe you can send us some data and we can look at it and we can say, aha, it looks like this, this, and this. We see a lot of data and we know how to look at it. We know how to interpret it, and we can tell you, I looked at this data and I saw, this, this, and this you might want to change. Or I don't see any problems with this data. Maybe you did just make a grand scientific discovery. And in that case, that is an excellent problem to have. So once you know your plant, and once you're keeping in mind that you need to note the survey, you need to know the theory and the reality for your plant. One category of measurements that people like to do is called survey measurements, or point measurements, or spot measurements, or quick measurements. Basically, what you're doing is clamping onto the leaf. You're seeing what it's doing at that moment in time, in those conditions, and you're going on to the next. You can do survey measurements once. You can do it over the course of a growing season. You can do it over the course of a day. If you do it over the course of a day, it's called a diurnal measurement. So you're going through the whole day. And then if you do it over the course of a growing season or a couple of growing seasons, then you can see the difference based on weather or the conditions or the treatments or the whatever. The other category of measurements is called response curves. In a response curve, what we do is we expose a leaf to a certain condition and we see how it does. So we say, I'm going to give this leaf lots of different light levels. What does it do? Lots of different CO2 levels. What does it do? Lots of different temperature levels. What does it do? That sort of thing. And this allows us to see how the plant behaves in different conditions. This is a much longer measurement in most cases. A survey measurement usually takes a couple of minutes per plant. A response curve typically takes 15 minutes to maybe multiple hours, actually. So response curves are much longer, but the idea is that you get different data out of it. The survey measurement is what is it doing right now? The response curve is what could it be doing, or what would it be doing if it was a different CO2 level, a different temperature, a different water, that kind of thing. The other thing you need to keep in mind when you're trying to think about your measurements is which chamber you want to use. Generally, in terms of chamber size, bigger is better if you can fill the chamber. The reason is that more leaf tissue means more flux. More flux means that we have more signal basically coming back from our leaf, either for fluorescence or for gas exchange. And more signal means that our signal to noise ratio is better. If we have, say, one balloon in a very large building, and we are trying to measure that one balloon, it's very difficult. We have very small signal. We have just one balloon, and we're trying to catch it. If we have a lot of balloons, they're easier to catch. So in this situation, if you have a very small leaf and a very big chamber, you have one balloon in a very big building and just trying to catch it, and it's hard. So what we want to do is, if we can, either make the building smaller or put more balloons in there. So if you can fill the chamber, use the biggest chamber that you have or that you can use. And the fluorometer has an option for aperture, so you can either do 2 square centimeters or 6 square centimeters. So that allows you to work with different sized leaves and not worry so much about filling the chamber or overfilling the chamber. a separate source of light, or are you just going to use the sun or the greenhouse lights or the growth chamber lights? So which do you need external light control? If you do, then certain chambers have it and certain chambers don't. And the other thing is, what data do you need? Do you need gas exchange and fluorescence, or do you just need gas exchange? We would encourage you, and one of the reasons that the multi-phase flash chamber, which is the one here on this slide, that's the default, because we learn so much more about the leaf if we have the option to do fluorescence as well as gas exchange, so we can tie those two components of the photochemistry cycle together, and we can get that information. We can do just gas exchange. We can do just chlorophyll fluorescence. If we put them together, though, that gives us much more information about what's happening in the leaf. And we always want more information about what's happening in our leaf, right? There are a few other chamber options for the LI6800. The multi-phase flash fluorometer, as I mentioned, is kind of the default, but there's also the red blue light source, which is a 3x3 chamber, 6x6 chamber, which has a red, blue, green, and white light source. We have a small plant chamber, a bryophyte chamber, a conifer sprig adapter, which means if you're working with short needled conifer type leaves, then you can go ahead and work with that. We have a soil flux chamber, an aquatic chamber for aquatic algae, an insect respiration chamber, and if nothing else, we have a custom chamber kit. So you build the chamber that you need based on the measurements that you need to take. Are there any questions about any of the theory that I have just tossed at you? That is the end of the theory lecture. It is just a, believe it or not, that is a brief overview of the theory of the LI6800. But are there any questions about that? Everybody understands exactly how this instrument works 100%?
ጠይቁ ግድ የለም ወይስ የቱን እንደሚጠየቅ ከየቱ ልጀምር ነገር ሆነ ጥያቄው? Is it too much information, too little, too detailed, or is it enough for now?
Okay, I have one question, but it's a very small question. When you said the measurement for stomatal conductance, you said we should avoid to measure the stomatal conductance when it's too dry or so too wet. In Ethiopia, especially on the summer season, it's cloudy usually. Sometimes it's no rain, also it's not dry, but cloudy, no sunlight. So in that case, can we measure the stomatal conductance in that period?
Yes, you can measure stomatal conductance when it's cloudy. The one thing you'll have to keep in mind is that you will want to decide if you want to use the ambient light or if you want to control the light. So if you have the leaf chamber fluorometer, it is an opaque chamber, meaning that there is not ambient light coming in, the sunlight doesn't come in. And then you would have to say, do I want to track the outside light, knowing that it's a little bit changeable, it's always changing? Or do I want to measure the outside light with my external quantum sensor, which is this little blue thing, which you will see a little bit more when we talk about hardware, and then set it to something near where that is. So